Hey, what's up, people? It's Movado 242, 12/15/2018. I don't know if you heard or not, a couple of weeks ago, this Chinese scientist or geneticist announced that he had successfully performed a DNA manipulation to prevent a baby from possibly contracting AIDS from one of the parents. Now, this was met with great consternation among the scientific community, the religious community, and the community in general, because, you start tinkering around with the DNA. We've seen all these movies where you have perfect babies. You can order the baby you want, the kid you want. And, it just leads down a question of, what's moral, what's immoral, what should we be doing, what shouldn't we be doing? Those are just some of the kind of questions that come up. But interesting, I noticed that that Chinese scientist that did that, last report I heard, he was missing. So anyway, and if the Chinese are doing it, we can't be outdone. The U.S. has to get their feet in the fire if they haven't already had it in there. And I would be very surprised if they haven't already. So this article comes from an author by the name of F. William Engdahl. He's a pretty good writer. I was reading one of his books, The Lost Hegemon, Whom the Gods Would Destroy. And on the cover of it, it has a silhouette of the United States and American flag upside down. So that should tell you a little bit about that. But it's a pretty good book. It takes a historical look at how we got involved in the Middle East and why we are there. Pretty eye-opening information. But this article he's writing in relation to the gene manipulation. And the headline, the title is U.S. Government Backs Dangerous New Genetic Manipulation Approach. And this article was written on April the 11th, 2018, so it's not new. The Trump administration is backing a new technology for the genetic manipulation of plants and even animals with no intent to supervise or regulate against possible dangers. If left unchecked, it could open a Pandora's box of dangers to human health for generations. Yet very little debate is taking place on this dramatic development. Here are some things to consider. The new development is being called genome editing, or simply gene editing. It is being promoted as a new, improved method of altering genetic expression of plants and even animals and humans. In 2015, London TED conference geneticist Jennifer Doudna presented what is known as CRISPR-Cas9. This is an acronym for Clustered, Regularly Interspaced Short Palindromic Repeats. It is a revolutionary and highly controversial gene editing platform using a bacterially derived protein, Cas9. It supposedly allows genetic engineers to target and break the DNA double strand at a precise location within a given genome for the first time. In effect, CRISPR is a highly precise way to alter genes in plants, in animals, and even humans, quite different from the older Monsanto gene canon or other techniques of changing a plant's genetics by bombarding it with a foreign substance to hopefully make the soybean or GMO corn resistant to Monsanto's Roundup weed killer, a highly sophisticated and highly costly procedure that is patentable and that is so complex as to be limited to a handful of company actors around the world. Gene editing is relatively cheap. Kits available for around $500. Wow. And relatively easy to use, as one analysis described it. CRISPR is a very precise, not to mention extraordinarily cheap and easy to use tool which can locate, cut, deactivate, activate, or rewrite any sequence of DNA that they want in a living cell. Note the words cheap and easy to use. Yeah, I was thinking about that. Can locate, cut, deactivate, or rewrite any sequence of DNA that they want. So we're going to have kids in their basement modifying DNA. Wow. At the cost of buying CRISPR and related genome editing materials, and the cost of buying CRISPR and related genome editing materials is alarmingly cheap, ranging from several $100s to several thousand. The equipment is available online from Scientific Equipment Maker, and on-site ad reads, CRISPR Cas9 editing made easy. Our easy-to-use, optimized, and validated solutions span the entire cell engineering workflow, making genome editing accessible to anyone at any level. And as one critic put it, anyone can buy some Cas9 for a few $100, and any halfway decent lab can use it to alter the DNA of anything. Wow. Man, what in the world are they doing? National security issue. DARPA, the Pentagon R&D arm, is spending millions on developing gene editing. In 2016, James Clapper, Obama administration director of national intelligence, added gene editing to the list of threats posed by weapons of mass destruction and proliferation. In July 2017, the Defense Advanced Research Projects Agency, otherwise known as DARPA, awarded $65 million in 4-year contracts to 7 teams of scientists to study gene editing technologies. The commitment officially made DARPA the world's largest government funder of gene drive research. So that's where your hard-earned tax dollars are going to. Gene drive is the next major development in the new gene editing world. The idea of gene drive is to force a genetic modification to spread through an entire population in just a few generations. Wow, man. Just think about that. The idea of gene drive is to force a genetic modification to spread through an entire population in just a few generations. Now that is scary. In the wrong hands, or in any hands, that is not good. One of the leading gene drive researchers, Omar Akbari at the University of California, Riverside, gets DARPA gene drive research money. He works with what is supposedly the world's deadliest creature, the Aedes aegypti mosquito. His work involves gene editing the deadly mosquito to re-engineering them with self-destruct switches. This sounds noble, a boon to mankind, saving maybe 1 million lives annually, like selling earlier GMO golden rice as a solution to infant blindness. Gene editing, however, is not as perfect as it is made out to be. The scientist who first suggested developing gene drives in gene editing, Harvard biologist Kevin Esvelt, has published a paper warning that development of gene editing in conjunction with turbocharged gene drive technologies have alarming potential to go awry. He notes how often CRISPR messes up and the likelihood of protective mutations arising, making even benign gene drives ruthlessly aggressive. He stresses, just a few engineered organisms could irrevocably alter an ecosystem. His computer drive simulations calculate that a resulting edited gene can spread to 99% of a population in as few as 10 generations and persist for more than 200 generations. It takes little imagination to conceive a scenario in which malevolent actors intent on wreaking destruction unleash destructive gene-edited plants or animals, even humans. Earlier GMO was so complex and costly it was limited to a very few actors who grabbed patents on their GMO seeds. Now with GMO editing readily available and becoming widespread, Pandora's box is opening wide. And the USDA government gives this the green light. Rather than approach such a potentially disruptive technology as gene editing with utmost caution and control, The U.S. government and the U.S. Department of Agriculture Secretary Sonny Perdue are opting for no regulation, a laissez-faire permissiveness that treats the resulting gene-edited plants as identical to conventional plants, therefore needing no special regulation. Now, just think about this. The FDA will go after people producing raw milk from their cows. They will bust down their door, raid their farm, and take whatever they have to take to prevent that from happening. But they're going to let this go with no regulation, period. Now, what kind of sense does that make? What does that tell you? They are not looking out for the interests of the people. In a March 28 press release, Perdue stated, USDA does not regulate or have any plans to regulate plants that could otherwise have been developed through traditional breeding techniques. This includes a set of new techniques that are increasingly being used by plant breeders to produce new plant varieties that are indistinguishable from those developed through traditional breeding methods, such as genome editing. So we have at the same time the U.S. intelligence community treating gene-edited organisms and related technologies as potential weapons of mass destruction, while at the very same time the agencies of the U.S. government responsible for food safety, the USDA and the EPA and FDA, seem content to turn a blind eye to all. As critics have noted, we might be able to wipe out an entire species on a whim. We might be able to do that to any species that we've decided we'd rather not deal with anymore, even undesirable groups of human beings. Wow, man. And that is incredible. I mean, the things that the FDA will ban that are, pretty much harmless or natural occurring. For instance, what was it? Keto, this drug keto, natural plant that could be used to get people off of heroin instead of taking methadone. They want to ban that, which is a natural plant, but they want to allow methadone. And this gene editing technology, which the paradox between it could be a weapon of mass destruction, but anybody can do it in their basement or in a decent lab with $500 or a couple of $1,000. You can start editing genes and creating plants and animals, and maybe you get some of your buddies over there and get them drunk. Next thing you know, you modifying their genes. Man, this is crazy. This is crazy. But the USDA says it's okay. They gave it the green light, so I guess it's good. It's good for the people. Yeah. Okay. Keep your eyes open, man, because this is this is not good. Anyway, have a good day and keep your eyes open. Peace.